Every moment of every day, trillions of cells in your body are making a critical decision, whether or not to divide. This seemingly simple choice determines everything from how a single fertilized egg becomes a complete human being to why cancer cells spiral out of control. Understanding this fundamental process remained one of biology's greatest mysteries until one scientist made a breakthrough that would change medicine forever. Sir Paul Nurse, a British geneticist and cell biologist, dedicated his career to answering a deceptively simple question: what controls when and how cells divide? In 2001, his work earned him the Nobel Prize in Physiology or Medicine, recognition for discoveries that revealed the molecular machinery governing one of life's most essential processes. The story begins with yeast, a humble single-celled organism that might seem far removed from human biology. Nurse chose to study fission yeast because these cells divide in a remarkably regular, predictable manner. By observing mutant yeast cells that divided at abnormal sizes or times, he could identify the genes responsible for controlling cell division. Through meticulous experimentation, Nurse identified a gene he called CDC2, short for cell division cycle 2. This gene produces a protein that acts like a master switch, determining when a cell has grown large enough and accumulated sufficient resources to safely divide into two daughter cells. The protein functions as an enzyme, adding chemical tags to other proteins, and thereby triggering the cascade of events that leads to cell division. What made this discovery truly revolutionary was what came next. Nurse found that the same gene existed in human cells, performing the identical function despite hundreds of millions of years of evolutionary separation between yeast and humans. This revealed something profound. The fundamental mechanisms controlling cell division are ancient and conserved across vastly different forms of life. The CDC2 protein works as part of a sophisticated timing system within cells. Think of it as a molecular clock that ensures cells only divide when conditions are right. The protein levels rise and fall in rhythmic cycles, and only when they reach a critical threshold does the cell commit to division. This prevents cells from dividing too early, when they might be too small or lack essential components, or too late, when they might grow too large and unwieldy. Understanding this control system illuminated why cancer develops. Cancer cells are essentially cells that have lost the ability to properly regulate their division. Mutations in genes like CDC2, or the genes that control it, can cause cells to divide uncontrollably, ignoring the normal checkpoints that prevent excessive growth. By revealing the molecular breaks that normally stop cell division, Nurse's work provided crucial insights into what goes wrong when those breaks fail. The implications extend far beyond cancer. The cell cycle machinery that Nurse uncovered plays roles in development, aging, and regeneration. When an embryo develops from a single cell into a complex organism with trillions of specialized cells, the precise control of cell division determines whether organs form correctly. When tissues need to repair damage, the same molecular switches must activate to produce new cells to replace those that were lost. Nurse's research exemplifies how studying simple organisms can unlock secrets relevant to human health. The fission yeast cells he examined in his laboratory are separated from humans by an enormous evolutionary distance, yet they share the same fundamental genetic toolkit for controlling growth and division. This unity of life at the molecular level means that discoveries made in yeast, fruit flies, or worms often translate directly to understanding human biology. The methodology Nurse employed, using genetic mutations to dissect complex biological processes, has become a cornerstone of modern molecular biology. By breaking the cell cycle and then identifying what was broken, he could work backwards to understand how the system normally functions. This approach has since been applied to countless other biological questions, from how neurons form connections to how immune cells recognize pathogens. Beyond his scientific contributions, Nurse has been a passionate advocate for basic research, the kind of curiosity-driven investigation that seeks to understand nature without immediate practical applications in mind. His work on yeast cell division had no obvious medical relevance when he began, yet it ultimately provided essential knowledge for understanding and treating cancer. This illustrates a crucial principle. The most transformative discoveries often emerge from asking fundamental questions about how the world works. Sir Paul Nurse's unlocking of the genetics of cell growth represents more than a single breakthrough. It exemplifies how patient observation, clever experimentation, and the courage to pursue fundamental questions can reveal the hidden mechanisms that govern life itself. The molecular switches he discovered continue to guide research into cancer therapies, regenerative medicine, and our basic understanding of what it means to be a living, growing organism. In revealing how cells know when to divide, Nurse illuminated one of biology's most essential processes, a rhythm that beats at the heart of all life.